Hi all, Mr. Duehr here. Today we're going to have a little discussion about enzymes because we're going to get ready for an enzyme lab. It's pretty exciting. So first of all, a quick reminder, enzymes are proteins. They're probably the protein we'll talk about the most in this class. And the specific type of protein that they're catalysts, right? They are catalysts, they're going to speed up reactions. And the reaction that we're dealing with today is the conversion of hydrogen peroxide to its products. You probably have seen hydrogen peroxide before. It's a typical first aid tool. It's often used for cleaning wounds. It doesn't actually sterilize like alcohol would, but it does help bubble out some dirt or bacteria that might have gotten into a wound, and so it can be used for that purpose. For reference, hydrogen peroxide only works when you have an open wound. So for instance, if I don't have any open wounds on my hand and I go and pour some hydrogen peroxide on my hand, nothing happens, okay? Nothing happens. There's nothing crazy that happens. It happens to splash all over my computer, which is really awesome. But other than that, nothing exciting happens. All that really happens is that it looks like water is pouring on my hand, right? But in fact, it's not water. It was hydrogen peroxide. I'm just going to tidy up here a little bit. All right. But what is amazing is that inside of our cells, and inside of many other types of cells, including most animal cells and even yeast and other kinds of cells, there's a special enzyme called catalase. You might have read about catalase a little bit. Catalase, as it so happens, is found in many different substances, and it's found in liver. And so here is a little chunk of cow liver. I bought it from the grocery store yesterday. Sometimes I buy chicken liver, but this time we bought cow liver. It works pretty nice. Chicken liver's pretty good too. So you have some options. Anyways, let's see what happens when we mix hydrogen peroxide with catalase. Do you think anything different will happen? Let's look. Whoa, that's amazing. White foam is forming. What could that be? Well, let's think about hydrogen peroxide and what its products might be when it gets converted. Hydrogen peroxide is H2O2. Its products are water and oxygen gas. Still going. So what do you think all those bubbles are filled with? Wow, it's really going. That's pretty good. What do you think those bubbles are? They're filled with oxygen. The reaction is definitely still going below. I can see production is still forming more oxygen. I probably put in too much hydrogen peroxide. That is outstanding. All right, we'll just let that diffuse for a moment so I can bring in that lovely smell. You guys are missing out. So that is catalase mixing with hydrogen peroxide. Now, the lab that we do is basically simulating that combination. We use, typically we take some catalase that's been blended, right? So liver juice, basically blended in with some water. Normally has a light pink tinge, but this year we're faking it with just plain water, okay? And we would normally use an oxygen meter. The oxygen meter connected to one of our vernier probe readers called a lab test stream. And this probe fits nicely into this chamber. And so in here we would put hydrogen peroxide, we would put some amount of enzyme, and then we could stopper it and then collect data for about 3 minutes. Okay, so we typically have this situation where we would be sloshing around, make sure that the enzyme is mixing and encountering lots of substrate, and we would go for a 3-minute time trial. And then we would repeat that over and over and over so that we had lots of data, lots of replicates for every different concentration. of enzyme and or for substrate concentration. Well, this year, we obviously can't do that in person. And so I have provided you with some data. And you have data for not only varying the enzyme concentration and how did that affect the oxygen production, but also varying the concentration of, or the amount of substrate, or how much hydrogen peroxide we have. And then finally, as an additional kind of thought experiment, we're going to look at how varying the temperature and the pH of the enzyme affects its productivity in converting hydrogen peroxide into oxygen gas. Do you think that these conditions might influence enzyme productivity? I guess we'll have to find out. All right, all. That's it for the moment. Let's get excited to do the lab.